Hello student, welcome to another lecture on Prokaryotes, Understanding Biology, Chapter 23. In this chapter, you'll be learning about bacterial genetics, including horizontal gene transfer methods and several important diseases and metabolic pathways of bacteria, usually prokaryotes. So at the end of this lecture, you should be able to describe how conjugation occurs in bacteria, transduction and transformation. These are different horizontal gene transfer methods. You should be able to explain and describe how these are occurring in bacteria. You should be able to compare different pathways that prokaryotes acquire carbon and energy and describe several important diseases caused by bacteria. So when you think about prokaryotic genetics, so in a sexually reproducing population, traits are actually transferred vertically from parent to child. Since you learned that previously, these prokaryotes do not reproduce sexually. So if that is the case, how do they exchange genetic material between different cells? So there is this process called horizontal gene transfer. So horizontal gene transfer occurs when genes move from one cell to another by these three different processes. These are actually observed in both archaea and bacteria. So all these processes are very common knowledge now, and understanding of these processes will help you to understand how prokaryotic genetics is working. So let's look into what are these three types of horizontal gene transfer methods. So we have conjugation, which is cell-to-cell contact. We are going to learn about it in a little bit. We have transduction, that is actually carried out by these bacterial viruses called bacteriophages. And lastly, we have transformation, which is acquiring DNA and genetic material directly from the environment. So without further ado, let's go into conjugation. So conjugation occurs when genes move from one cell to another through cell-to-cell contact. So plasmids that we talked about, that bacteria, other than their chromosomes, they have plasmid. So this plasmid may encode these functions that are advantageous for prokaryotes. In some cases, this could be like added advantage because of the antibiotic resistance. So these plasmid actually can be transferred from one cell that containing that plasmid to another cell that does not have the plasmid via a process called conjugation. The best known transmissible plasmid is called F plasmid, or it is also called a fertility factor. So we usually call it F plasmid. So yes, you can see the cell that has the F plasmid are called F positive cells, and the one that lack the F plasmid are called F negative cell. When F plasmid, when we look at the process, how the F plasmid is transferred, it is actually quite complicated. So F plasmid in E. coli actually contain this DNA replication origin and several genes that promote its transmissibility or transferring to other cells. So the plasmid itself actually encodes genes that allow it to be transferred to other cells that are F negative. So first what happens is that these F plasmid, these genes will encode protein subunits that assemble in the bacterial cell surface. So the first thing that need to be done is that it need to produce a pathway where the plasmid copy can reach to the other cell. To make that pathway, the the plasmid genes will encode proteins that assemble on the bacterial cell surface, forming this halo pilus, which is very necessary for this process. So after they started making this halo pilus, this F1 plasmid actually bind to the site on the interior of this F plus cell just beneath this pilus. Now this pilus is called the conjugation bridge, as you can see here. Once this plasmid bound to the interior of this pilus is called now the conjugation bridge. So then F plasmid start to replicate. I told you that they have DNA replication origin, and they start replicate, and it is actually replicated, this process called rolling circle replication. So a single stranded copy of this replicated plasmid will be transferred through this pilus to the other cell, which is F negative here. So once this single stranded copy is transmitted to the F negative cell, they will actually make the complementary strand and complete the process, producing a new stable F plasmid to the one that didn't have. Now both of these cells will have F plus plasmid. So now after the cell receive this F plasmid, there are several things that can happen with the F plasmid. So one of the things that can happen to the F plasmid is that it can actually integrate into the host chromosome through this process called homologous recombination. This homologous recombination occurs through finding out these homologous regions of DNA present in bacterial chromosome, and these homologous regions in E. coli chromosomes are called integration sites. Once they find the integration site, that matching will make a complementary base pairing, and the F plasmid can be integrated into the bacterial chromosome. A cell with integrated F plasmid is called HFR, for high frequency recombination. Now F plasmid transfer will also include chromosome DNA sometime, because now that F plasmid has integrated into the chromosome, they will start replicating every time the host is dividing, and it can actually excise itself, reversing the process, and it can carry some of the bacterial chromosome DNA with it when it actually excising itself. So this integrated F plasmid, actually there is a process that actually help it to excite itself from the chromosome, and sometimes through excision, inaccurate excision can happen that allow them F plasmid to pick up some of the chromosome DNA, and then it will call F dash plasmid, because now it has some of the chromosomal DNA. Now you can kind of understand how bacterial genetic diversity is happening, because they are mixing, even though they don't have the sexual reproduction. Through this horizontal gene transfer, they will have the some of the mixing of genetic material between different bacteria. So in this picture is actually showing the integration and excision of F plasmid to the bacterial chromosome. In the integration, you can see that F plus cell, once it get this plasmid, it will find these integration sites which are homologous to each other. They will make complementary base pairing and actually bind this F plasmid to the chromosome. Then it will become HFR cell. Now if it want to reverse the process and excite this F plasmid back again, then it will actually go through this reversal process where it will break this plasmid out, which if it actually gets some of the bacterial chromosome DNA, it will become F dash. If not, if it accurately excision happens, it will again become F plus cell. So when a conjugative plasmid pick up resistant gene, it is called a resistant plasmid. So I told you that sometimes inaccurate excision can actually carry some of the chromosomal DNA with the F1 plasmid, and once the F plasmid gets these resistant genes, these plasmid are called resistant plasmids. So resistant plasmids are actually very advantageous for the bacteria that is harboring them, because it will allow them to survive in the in the presence of antibiotics. So when they transfer this R plasmid to another bacteria, they also actually get the antibiotic resistance, and they also can transfer genes involved in virulence. Virulence is the ability of bacteria to cause diseases. And especially these are called virulence plasmids, plasmids that actually have other genes that actually allow them to become more virulent. So they actually encode genes for pathogenic traits, and especially bacteria of Enterobacteriaceae have this virulence plasmid, and E. coli this E. coli strain cause hemorrhagic gastroenteritis, which is also having this virulence plasmid. So now that we are done talking about conjugation, let's look into another horizontal gene transfer method called transduction. So this horizontal gene transfer can also be mediated by these viruses infecting bacteria called bacteriophages. Bacteriophages can transfer bacterial DNA in two ways. First one is called generalized transduction, and the other one is called specialized transduction. In generalized transduction, any gene can be transferred between cells. There is no specification. In a specialized transduction, only a few specific genes can be transferred. So let's look into horizontal gene transfer method, generalized transduction. So generalized transduction occur mainly in lytic phages. So you've learned about two different lifestyles of phages, lytic and lysogenic. Lytic phages actually replicate inside the bacterial cell, lyse the cell by killing it, and release new virions. And this generalized transduction happen during the lytic life cycle. After viral genome is replicated, these phages sometimes accidentally pack bacterial DNA instead of the viral DNA. So when this bacterial DNA carrying phage goes to another bacteria and inject its DNA, this DNA is actually bacterial DNA instead of viral DNA, so it won't cause the infection, but this DNA can be incorporated into the recipient chromosome by homologous recombination, and this will actually give certain non-specific genes, because this process occurs randomly. And the other method, which is on the other hand the specialized transduction, happens only in lysogenic phages. And you know, these lysogenic phages, they usually infect, but they do not kill the host unless it is necessary. They will integrate their DNA into the host chromosome and then replicate through the host chromosome with the host chromosome until the environmental conditions are not good. Then they can become lytic and lyse the cell and go find another host. In this life cycle, what happens is that when a phage integrate into the host genome, they can actually reverse this process and excite, right? So once they integrate their genome into the host genome, host chromosome, they are called prophages. When prophage is exciting itself from the host chromosome, sometimes imperfect excision can take some of the bacterial chromosome and DNA with it and introduce it to another bacteria. But it is very important to notice that in a specialized transduction, transducing phage can carry only the genes that are adjacent to integrated site. This is why it is not very randomly happening. It is very specifically happening. Only the genes that are near to the integration site can go through the specialized transduction. Let's look into a picture of the generalized transduction here. This bacteriophage is actually carrying the bacterial DNA instead of their own DNA. They inject the DNA, which is like homologous sites can find through the homologous recombination and integrate into the host chromosome. And the next horizontal gene transfer method we are going to talk about is called transformation. And this process is where cells will uptake DNA directly from its environment. This is actually a naturally occurring process in many bacterial species, and especially if you remember the experiment done by Frederick Griffith, the streptococcus studied by him, he noticed that the transformation occurred when one bacterial cell died and ruptured, spilling its DNA into the environment, and the other bacteria actually took up this DNA from the environment through this transformation and incorporate it into its genome by homologous recombination. There are actually proteins involved in the process of natural transformation that are encoded by bacterial chromosome. This is not any kind of accidental process. This is very much planned process, because bacterial chromosome actually producing these proteins that involve in the process of transformation. So here is a picture of a cell death of a bacterium spilling its pieces of DNA into the environment, and here is a recipient bacteria that can be naturally transformed. It actually absorbs this DNA from the environment and integrate it into its own genome. And some species of bacteria are actually not naturally receptive. When transformation is actually accomplished in the laboratory, this is called artificial transformation. So sometimes, if you know, in molecular recombination, we use E. coli for molecular cloning to introduce these genes of that we are interested in into the plasmid and transfer that plasmid into a bacteria that can produce the product of that gene that we are interested in. We can study them. So now I think you have a good understanding of horizontal gene transfer methods of prokaryotes. Now we are going to talk about prokaryotic metabolism. One of the major reaction or reason for prokaryotics diversity is its biochemistry rather than morphology. There are many different types of metabolic pathways among prokaryotes, including how they acquire carbon and energy. So based on how organisms acquire carbon and energy, we can categorize them into two main groups, which you already know, autotrophs and heterotrophs. So these autotrophs and heterotrophs are further classified into four metabolic lifestyles. These are protoautotrophs, chemolithoautotrophs, protoheterotrophs, and chemoheterotrophs. So protoautotrophs, they acquire their energy from the sunlight and convert carbon dioxide to organic form. On the other hand, protoheterotrophs, they do use the light energy, but they actually obtain their carbon from organic molecules such as carbohydrates and alcohols. Protoautotrophs actually convert carbon dioxide to organic form. They fix carbon dioxide. But protoheterotrophs, they actually obtain their carbon from organic molecules such as carbohydrates or alcohol. And we do have chemolithoautotrophs, which they obtain energy by oxidizing inorganic substances. They can oxidize ammonia or sulfur, but most prokaryotes are actually chemoheterotrophs. They obtain both carbon and energy from organic molecules. Humans are also an example of chemoheterotrophs, and many of the strains of bacteria that we culture in the laboratories, in microbiology laboratories, that growing in media, they are also chemoheterotrophs. So now that we have a good understanding on horizontal gene transfer methods and prokaryotic metabolism, let us conclude this lecture by discussing diseases caused by bacteria. So in early 20th century, before discovery of antibiotics, infectious diseases actually kill nearly 20% of all children before they reach the age of 5. Actually, finding and discovering of sanitization and antibiotics actually considerably improved this situation. And we already found out that in recent years, however, many bacterial diseases have appeared and reappeared because of this multi-site drug resistancy. Actually, bacteria are becoming more resistant to the antibiotics that we have recently. So it's very important for you to have some understanding on what are some of the most important diseases of bacteria caused in humans. So in this table 23.1, you can find out some of the important human bacterial diseases. So there are many different diseases caused by bacteria because they have different methods to spread through the susceptible population. Some of the spreading methods include spreading as air droplets of mucus and saliva, fecal contamination, or using different vectors such as tick vectors. So some of the important diseases include anthrax, botulism, chlamydia, cholera, dental caries, diphtheria, gonorrhea, human disease, leprosy, Lyme disease, plaque, and all of that. You can see tuberculosis. So all of these diseases, as you can see, some of them infect your gastrointestine, some of them infect your oral health, some of them are actually sexually transmitted. So these are actually some of the important diseases caused by bacteria in humans. So let's look at some of the diseases. So the first one we are going to look at is tuberculosis, which is second only to HIV or AIDS as the cause of death worldwide. This disease is caused by this bacteria called Mycobacterium tuberculosis, which affect the respiratory system. They can actually prevent the immune system from attacking them and preventing the disease, and they are easily transmitted from person to person through air. They also have multi-drug resistant strains, which is very alarming because there is a possibility that tuberculosis will be another outbreak in future. And we also know that many bacteria can cause gastrointestinal disease, and they can also live in our oral cavity. So one of the diseases caused by bacteria in gastrointestinal tract is called peptic ulcers. This is due to these cater-like lesions in the gastrointestinal tract that are exposed to peptic acid. This is actually previously we thought because of these different kind of reasons such as cancer, these peptic ulcers are caused by them. But actually later we found out that it is actually caused by Helicobacter pylori, which is a bacteria. And now we can actually treat it with the antibiotics to cure the peptic ulcer disease. And another disease caused by bacteria is called dental caries or tooth decay, which you are very familiarized with if you have plaque. Not all the bacteria that are in the plaque are very bad. Some of them are actually good. But there are two main bacteria that you have to watch out for in plaques. Plaque is actually a biofilm caused by different species of bacteria. And these two species, such as Streptococcus sorbimus and mutans, are the most important in tooth decay disease because they can actually ferment sugar to lactic acid. So once you eat sugar, they are actually very bad because soon these bacteria that is in the plaque will convert the sugar into the lactic acid. Lactic acid actually can degrade and eat your teeth's enamel, which actually degrade the teeth over the time if you do not clean your plaque out with dentist. So make sure you floss your teeth every time to prevent these plaques from forming and having your teeth degraded. So that is my last slide. And in summary, today you learned that prokaryotic DNA exchange is horizontal from donor cell to recipient cell. DNA can be exchanged by conjugation via plasmid. The DNA can be exchanged via transduction by viruses, and DNA can be exchanged by transformation through the direct uptake of DNA from the environment. We also learned that prokaryotes have both autotrophic and heterotrophic species, and you learned about 4 different life cycle or lifestyles of metabolic lifestyles of prokaryotes. And we did also learn about many human diseases, including tuberculosis, streptococcal and staphylococcal infection, and sexual transmitted diseases are caused by bacteria. So I hope you enjoyed this lecture, and see you again in another lecture. Thank you.